Well, good morning, and welcome to Grand Challenges. I want to thank Prime Minister De Croo for being our host here in Belgium. Our foundation is proud to have a strong partnership with the government of Belgium, particularly focusing on African sleeping sickness. It's making a lot of progress towards elimination of that disease. I also want to thank President von der Leyen for joining us today, but also for her leadership in global health. Just last month, when we had the Global Fund fundraising, with her leadership, the European Union increased its contribution by over 30%. Also, during the pandemic, where many countries were taking fragmented approaches to COVID-19, the European Union pulled together to mobilize not just a response for the European Union, but for the world as a whole. Our foundation has a number of great partnerships with the European Union, including areas like genomic sequencing and regulatory systems in Africa, polio eradication, and trials for new malaria and TB drugs. Lastly, I want to thank everyone involved with Grand Challenges who's here today. When it first started back in 2003, we knew partnerships would be a key to supporting great science. But we didn't realize that we'd be able to create so many different Grand Challenges programs. The first of those was Grand Challenges Canada. So they continued that on, doing great work, and now we have many others, including the latest, which is Grand Challenges Pakistan. And so it's not just a Gates Foundation movement. Yesterday, right here, some of you participated in the pandemic simulation exercise. And I thought it was fantastic because all the complexities of decision-making really came out during that exercise. I think it's clear that we need, at the global level, a team of experts that I think of as like a global health emergency corps to be ready to practice and make sure that next time we do a far better job. I'm hopeful that we won't forget the lessons of this pandemic before we actually build the infrastructure that will help us be ready for the next one. COVID-19 challenged us in many ways, not just in terms of the pandemic preparedness, but also in terms of how we do all of our product development, bringing a new sense of urgency and perhaps showing us that we can get products out faster than we did before. We treated everything like an emergency. We got sequencing tools out, we got oxygen out, we didn't let business as usual hold us back. And many of the diseases that Grand Challenges focuses on, the ones that are incredibly inequitable, HIV, diarrhea, malaria, NTDs, tuberculosis, the burden of these diseases over the last decade is very similar. They're an equivalent tragedy to the number of deaths caused by the pandemic. So we ought to really re-examine in all the tools work we do exactly how we can accelerate things. One of the most important things was diagnosis. In fact, if you look at the different results in terms of the death rate of different countries, one of the key differentiations was that some countries didn't just stay with public health labs, they went out and got commercial PCR capacity, and so they were doing population level testing early in the pandemic. That was a gigantic benefit. Right now, diagnostics are not very available, and so maybe we should think about changing the whole paradigm. For example, as much as possible, we should avoid having to go to a health facility. If you can administer the test accurately in your own home, then we can catch your infection way earlier. And that's particularly important if some of the drugs or other therapies only work at the early stage of the disease. So we ought to really consider bringing tests to people, and not just for COVID, for all of these diseases. The foundation has a partnership with BioCrucible and Flextronic to come up with very inexpensive lateral flow type testing that could fulfill this dream. Now, you would do the sample acquisition with things like tongue swabs or nasal swabs. We have partners that are developing the latest versions of those, and then you would simply put it into the test yourself. For some tests, you may still have to go to a health post where they have a machine for things like blood tests and other tests. And now some of these machines are getting dramatically less expensive and requiring less training. For example, the Lumira machine, whose tests will span all of the things we want in health posts. So if we can take either the tests that run through the machine or the home tests and literally make 16 million on a single manufacturing reel in a day, that's a level of scale up that would work to help stop an outbreak from becoming the next pandemic. Given the extremely low cost there and the fact that we can adapt that manufacturing to new tests very easily, it also means, whether it's the rich world or developing world, we can have a lot more testing going on, catching malaria cases earlier than we do today. We also think that central lab testing, a new version of what was the commercial PCR that was fairly expensive, that there's an approach that actually comes from the field of agriculture in terms of how they test the genetics of seeds, and it can be 20 times less expensive and 20 times more efficient. And that's actually running today in New York and Johannesburg. So whether it's at home or at the first healthcare post or the central lab, the quality and cost of diagnostics, both for pandemics and regular diseases, we think can be revolutionized and have far more access. Moving on to vaccines, the most spectacular breakthrough for the pandemic, of course, were vaccines, in particular the mRNA vaccines, but not only those vaccines. The mRNA approach does have some huge benefits in terms of it's one standard manufacturing approach. And so if you want to make changes, you're just changing that mRNA message piece. So in terms of cost and time, it's very dramatic, the benefit you can get. What's missing? There are three characteristics that even the mRNA vaccines aren't giving us today. One is duration. We'd like to have lifelong protection so they don't have to go back and be boosted again and again. There are approaches to solving this, including circular RNA. A second area we need to improve is breadth. We need to make sure that every new variant doesn't require us to readapt the vaccine. As we understand immunology better, and actually even the dosing rate, that where you have continuous exposure instead of one-time exposure, that the breadth of antibodies can be far better, I think we can make breakthroughs on duration. The final characteristic that we're missing, and I'm sure many of you have experienced this, is that just because you're vaccinated doesn't mean that you can't get infected. We hope that these vaccines would create the mucosal protection and give us more community immunity, but in fact we got very little of that. It looks like delivering mRNA in an intranasal form will give us some of that mucosal protection. And so already tests are being done of that. So over the next few years, we may be able to solve that problem. In the whole area of drug and vaccine manufacturing, we also need innovation. We saw that we had a lot of dependence on a few locations, and we couldn't scale up the manufacturing. Well, back in 2016, we made a Grand Challenge grant to a startup company called Cells. It's headquartered about a half hour from here, and they had a new approach for doing vaccine manufacturing in a kind of modular way that would allow it to be done in far more locations. They combined several processes into one. They made it so that it's very easy. The skills required for running the manufacturing facility were much simpler, and it wouldn't require much space. And so in collaboration with Batavia Biosciences, Cells has already taken this approach and made it work for a variety of vaccines, in particular measles, rubella. And they recently reached an agreement to apply this technology in Senegal at Institut Pasteur Dakar, and even the initial factory will be able to make 40 million doses. And so that's really the start of distributed manufacturing for all of these tools, making for a more robust supply chain. The last area to talk about, of course, is drugs. Drugs have actually had a very modest effect in terms of lives saved in this pandemic. You know, we did get some great antivirals like Paxlovid, but because it requires testing early on to have an impact, it hasn't been that dramatic. And in fact, if we look at how the trials were done for the various drugs, there wasn't the level of collaboration and trial design to both eliminate false leads and help us understand what the best drugs were efficiently. You know, so unlike the vaccines, this was not well coordinated, although a few tools like dexamethasone did come out at a fairly early stage. We've been working on a collaborative model for drug development for many of the key diseases. And I want to share with you that I think that's going extremely well, and it's kind of a model that I'd like to see us use for even more of our drug pipelines. So I have examples from both our TB work and malaria work. In 2012, we launched the TB Drug Accelerator. It's now got 24 partners, including academia, pharmaceutical industry. In 2017 was when we did the same thing for malaria, and that's got 17 partners. We don't just have diverse types of partners, but we also have partners from high-income countries and from middle-income and low-income countries as well. Now, traditionally, everybody would have worked in their own silos, and the fact that one partner had a certain skill that they were better at and another one had a different one, we wouldn't get the benefit of that. But under the accelerator framework, we have complete sharing of the compounds, the data, and that expertise. And so if one organization is good at X-ray crystallography, another one is using machine learning, we get the full benefit of all of that expertise. To give an example coming from the TB drug portfolio, a team from Cornell developed a screen for compounds that kill the bacterium while it's in its latent state, hiding away in host macrophages. Researchers at another partner, Caliber, which is part of Scripps Research, saw that they had some hits, but they wanted to modify the structure. And using information that GSK offered from its research, we were able to see that we had the right mechanism And so it really took all of these different partners coming together to come up with this drug candidate that looks very, very promising, particularly in this case for reducing the number of months you need to take the TB drug regimen. And that, that's a huge problem. You often have people quitting early, and then not only do they get infected again, but they're infecting other people. So, Grand Challenges is here to drive any innovation that helps us save lives. We measure the work here by health and equity, reducing deaths, reducing morbidity, and giving people the promise of a healthy and productive life. The Grand Challenges movement has more smart people from more countries than ever, and so I'm very excited that the understanding of how to design drugs, how to deal with things like the microbiome, many deep understandings that should help us increase this rate of innovation, particularly taking the lessons and urgency from the pandemic. We do have to continue to always have that metric be full delivery, not just discovery. The science is great, but understanding the challenges, whether it's cold chain or trust, that reduce the acceptance of these tools, we have to be very, very smart about that as well. So more urgency, more innovation, more partnerships, I think should make us all optimistic about what we can do. So the years ahead, we have a chance to save millions of lives. Thank you for your partnership in this great endeavor.